Hi, my name is Chanelle Adams, and I'm Senior Application Specialist at Roca Bioscience. For the next couple minutes, I would like to give a brief talk on ways of overcoming the challenges of pathogen testing in difficult foods. So a few of the subtopics that I would like to discuss will include what is a difficult matrix, test method validation versus verification, and lastly, rapid detection of pathogens in such difficult matrices. So the first issue of resolve will be for us to identify what exactly constitutes a difficult matrix. We oftentimes hear this term, but are we certain of what this encompasses? Working with customers in the food industry, oftentimes we are asked of ways how to successfully identify pathogens in these challenging or difficult matrices. So a few of the matrices that I've encountered in my experience that serve difficulties in successfully identifying pathogens and analytical testing methods include things such as spices, chocolate, dry pet food, or even whey protein powder, to name a few. Some of the reasons why these pose threats or challenges to these testing platforms are that they can attribute characteristics such as growth inhibition. Will the microorganism be able to grow to detectable levels in such matrices? It can also interfere with assay detection due to such things such as pigmentation. One of the matrices mentioned included spices. If we take, for example, a spice such as paprika, the bright red color, the pigment can interfere with PCR detection or other assay methods. And another area of concern could also be high background flora. So these are some of the things that we encounter that could serve challenging for analytical testing platforms. So one of the greatest areas of concern in the food industry is the potentiality of having to put forth a recall on a product. And this can be because a potential pathogen was not correctly identified, or it could be the result of a false result. If we take into consideration one of the difficult matrices presented on a previous slide, let's say whey protein. If a company was interested in identifying pathogens such as salmonella or listeria in the finished product of whey protein, there are certain things that must be taken into consideration. One, with the matrix of such composition, there's a high potentiality that the particulate can transfer into the reaction, and this is what we would consider a matrix effect. This attributes physical interference into the reaction and could, one, either inhibit the method of interest or two, produce a false result. In regards to chemistry-based inhibition, we can use a such matrix as chocolate. Chocolate contains polyphenolic compounds, and because of its composition and formulation, there's a high lipid content. These can also interfere with downstream PCR reactions. In regards to high background competition, these things can affect enrichment efficacy, and there's also a likelihood of introducing cross-reacting organisms into the analyses. So even though these challenges exist, there are ways of overcoming them. And oftentimes when a laboratory is interested in testing a product, they want to find the most accurate result, but at little cost. There are ways of overcoming these challenges, which may include, one, either upfront sample purification. So is there a way to clean the matrix of such interference substances so that we can get the most accurate result in the shortest amount of time? If we take upfront sample purification into consideration, over the long haul, this could eventually extend the time to result, and it's also an additional cost. There's a possibility of extended enrichment, and again, this could extend the time to result. But for a lot of these matrices that serve difficult for several testing platforms, special media or supplements are often needed. Laboratories aren't necessarily aware of this if they are not performing in-house testing. So when a third-party laboratory contacts them and informs them that the cost of testing may need to be increased, they oftentimes don't have an understanding as to why. In a few slides, we'll get into matrices that may call for the addition of special supplements or variations in media. So again, some laboratories may try to avoid added costs as long as a third-party laboratory or their very own laboratory can identify potential foodborne pathogens. The Centers for Disease Control reports that annually in the United States alone, approximately 48 million people, that's 1 in 6 Americans, will get sick, 128,000 are hospitalized, and 3,000 will die each year from foodborne illness. And so as a result of this statistic, the government has begun to try to shift the mindset from reacting or responding to contamination of our food source to actually preventing it from happening. And the way that this is being conducted is through the Food Safety Modernization Act. So for the first time in approximately 70 years, the shift or the implementation, of government regulation will shift the mindset from a comprehensive approach to that of being more scientific based to prevent contamination of our food supply. The Food Safety Modernization Act will also require preventive controls with verification across the food supply, which correlates to facilities needing to have a written food safety plan that clearly states a verification testing program. So overall, pathogen testing will function as a verification of the effectiveness of preventive controls. So the second subtopic that I wanted to introduce is that of validation and verification from a test kit manufacturer standpoint. Most food companies are familiar with the hazard plan and are therefore familiar with the term of validation. But when we're looking at it from a test kit manufacturing standpoint, there are different areas that will require attention. So validation is performed to demonstrate that the method is equivalent to the reference method based on defined method criteria. And on this slide, we see that method is in bold. And what I'm trying to get across here is that the method emphasizes the analytical testing platform that is being utilized. Whatever method of choice is selected, it has to be equivalent to something that has been set forth by either the Food and Drug Administration or the USDA. So several foods pose different challenges in their complexity. And the complexity of food matrices is what poses this significant challenge in laboratories adopting molecular techniques. Again, a lab wants to be able to successfully identify a pathogen or a potential pathogen in their food product, but they also want to take into consideration the cost that's associated with it. So in performing a validation study, a company will be most interested in identifying the platform that can accurately and confidently identify these pathogens without the likelihood of presenting false results. So as mentioned in the definition of a validation, whatever test method is of interest for a laboratory, it has to be equivalent to a reference method. Now, food projection requires highly sensitive pathogen detection, but once again, the complexity of these matrices can pose challenges to adopting molecular techniques. There are specific developments in sample preparation as well as assay technologies that are becoming more prevalent in identifying ways of overcoming these challenges. And when I say that a test method must be equivalent to a reference method, I'm placing emphasis on the fact that whatever culture method is utilized, the result has to match that of what has been mandated by either the FDA or the USDA. So in selecting these analytical testing methods, the method should be able to confidently detect and identify the analyte. And there are certain characteristics that must be taken into consideration. Is the assay or platform of choice sensitive? If in a laboratory I'm interested in identifying salmonella, will it only produce results for salmonella? Or is there a likelihood that E. coli, Klebsiella, Citrobacter, or any of its distant relatives could pop positive results on a platform of interest? It also must be specific. Once again, if I'm interested in identifying salmonella in a product, will I only detect salmonella and not listeria? We're also interested in identifying the accuracy, reproducibility. If I'm running 2,500 samples bi-monthly, will I confidently be able to identify this pathogen of interest? And lastly, is it fit for purpose? Is this platform that I'm interested in the best platform to identify my needs? So AOAC produces food categories that must be considered in validating such matrices. On the slide before you, I've presented 4 separate categories: meat products, fruits and vegetables, chocolate or bakery products, and a miscellaneous category. When a test kit manufacturer is interested in validating a specific matrix, that matrix would have to fall under one of these food categories. I've highlighted on the slide milk chocolate as well as spices, mainly to place into our memory that these products have been categorized as difficult matrices. So once a category is validated, let's take for instance, chocolate or bakery products. If I'm in a laboratory and I have validated milk chocolate, my claim of validation will solely be limited to that matrix that I've actually tested. So just because I've validated my platform for milk chocolate does not mean that I have the ability to lay a validation claim to, let's say, a poultry product or a raw meat product. So the study design that would be of interest or that would be required, by the AOAC for a test kit manufacturer would be to include an inclusivity panel, and this is another way of saying that the assay or platform of interest is actually sensitive enough to detect the organism of interest. I would also, as a test kit manufacturer, have to include an exclusivity panel, and that means that the organism that could also be present in a product does not interfere with that target organism. A method comparison will have to be performed, and there will also have to be evidence of robustness of such a platform. So if we take into consideration an inclusivity panel, what that means is that between 30 to 50 strains of a specific organism would have to be tested on the platform, and the platform successfully identify it. Salmonella is a special case because of its extensive serovars. The inclusivity panel for salmonella must include 100 strains. The method comparison would have to be compared to either the USDA, FDA, ISO, or any of these other reference methods. So whichever result is produced by their recommendations, the test method should produce the same result. So what does a validation study include from a test manufacturer kit standpoint? In a customer applications laboratory, when we're conducting a validation study for a customer, we'll typically design it so that it includes 30 samples in totality. These 30 samples are divided into 3 groups. The first being 5 non-inoculated. We'll take the matrix of interest, not spike it with any organism, enrich it in alignment with the remainder of the samples, and test accordingly. 20 of those 30 samples will be inoculated at a low level, and that's targeted between 0.5 and 1 CFU. So what we would try to show here is that even at extremely low levels, with a very limited likelihood of an organism being present, that the assay or platform of interest, again, has the sensitivity required to prevent the likelihood of producing a false result. And the last set would include a 5 subcategory of highly inoculated samples. For this group, we'll spike the sample or the matrix of interest to target between 6 to 10 CFU per gram. This will not overwhelm the system, but again offer confidence that a pathogen or microbe of interest could be detected. So a few slides back, I mentioned some of the pros and cons or some of the difficulties that laboratories must encounter when identifying costs associated with selected or identifying the analytical testing platform that is of best interest to suit their laboratory needs. One of the caveats to overcoming identifying pathogens in difficult matrices was to, one, either utilize special media or to, secondly, introduce supplements. On the slide before you, I have an example of onion powder. Spices typically have the ability to present challenges antimicrobially to organisms and thereby interfere with their ability to grow. And so ways of overcoming that either present challenges of performing dilution or, secondly, utilizing additives. In the case of onion powder, a specific additive is required in the set of potassium sulfite. And by introducing this, it serves to inhibit the antimicrobial property of the onion powder, allowing the organism the opportunity to thrive and grow to detectable levels. The second parameter that can be performed by a test kit manufacturer is to perform what's known as a verification study. And verification studies are performed to demonstrate that once a platform is in the hand of the user, that specific method can be performed to meet the defined method criteria. So in performing a validation study, as we just walked through, we were more interested in identifying the ability of the method, that analytical testing method of interest, and its ability to successfully identify the target organism. But in performing a verification study, the interest is switched from the platform itself because we've already determined its ability to identify the organism to can the user or the laboratory or the company that's interested in testing the method, do they have the ability to implement a testing parameter that can aid in successfully identifying a potential pathogen. And so a verification will be performed if that specific matrix of interest falls into a subcategory of a validated class. So if we think back a few slides where I showed the AOAC validated categories for food classes, we had meats, we had miscellaneous, chocolates. And so if I was interested in testing something that fell into one of the same categories, I wouldn't have to walk through an entire validation study all over again. Instead, I could perform a verification. And this is what's shown in the second bubble, the blue bubble on the right, that if I have a new matrix or even a new target organism, I wouldn't have to walk through a validation study all over again. So let's take, for example, that chocolate that was highlighted in the food category. If I'm a laboratory who is currently validated for milk chocolate, but I want to begin manufacturing dark chocolate, both of these matrices will fall under the same food category of chocolate or baked goods, and therefore a verification would be applicable for implementing the same testing methods. So again, both dark chocolate and milk chocolate fall under the same food category. The only difference that is exhibited amongst the two is that there are differences in product formulation. The milk chocolate may have naturally occurring antimicrobials, which can inhibit microbial growth, but because the laboratory has already performed a validation study on dark chocolate, they can present the ideal that there is a strong likelihood that the two products will behave similarly during testing. So verifications are performed when a new matrix is a subcategory of a validated food category. And in the customer applications laboratory, verification studies are typically designed as what we refer to as a 7x7 study. This will include 7 non-inoculated samples and 7 inoculated samples. These 7 inoculated samples are targeting a high level, so somewhere between the 6 to 10 CFU per gram, and the non-inoculated samples will endure the same enrichment as those that have been inoculated. One of the benefits to this is that it is smaller than a validation study, so not as many samples are required. However, the confidence that this method will succeed has already been established by performing the validation on a major category under the same food class. So lastly, I would just like to walk through things that should be taken into consideration for rapid detection techniques. There are several platforms that are out in the industry, and several people may have recommendations. But what's of most importance is identifying a detection method that serves the greatest purpose for your interest. A lot of times customers inquire about the robustness of a platform as well as the turnaround time. If you're in a manufacturing plant, one of the hardest things to do is to continue to manufacture products and not have anywhere to store it because we're waiting on results. It's also important to look into whether or not a platform has been validated for specific matrices, because we have to keep in mind that not all detection methods are the same. So if we just think about the way that technology has advanced over the years, there are certain things that need to be taken into consideration. The enrichment method that the FDA and USDA typically rely on, which would be our reference method, and which the test method must be compared to, is that is culture-based. And this idea of culturing organisms for isolation evolved around the 1880s. And although it has served useful over the years, it requires a 5 to 7 day turnaround time for a laboratory to obtain results. That organism of interest must be identified, proper media prepared, plates poured, or they may be purchased pre-made. The sample must be enriched or grown overnight so that detectable levels are present, the cells are healthy and viable, and then those samples must, after incubation, be streaked onto a selective or differential media so that it can be confidently identified. After streaking to such media, those plates must be incubated and 24 hours later read, and certain biochemical tests must be followed up to confidently identify that organism. If we fast forward to somewhere around the 1980s, we see the advent of immunoassays. It's faster than culture, but it still requires approximately 2 to 4 days to obtain the result. Immunoassays are sometimes subject to cross-reactivity as well, so in attempting to identify the sensitivity, these assays could serve problematic. We fast forward to the 1990s. We see the advent of PCR by Carrie B. Mullis, and this, again, is faster than immunoassays. So as we travel through time, we're seeing a quicker turnaround time. But one of the downfalls to that is, again, it could be 2 to 3 days till you get a result. And PCR also shows that it's subject to interference substances. So this can be high background and also lead to the possibility of either identifying invalids or producing false results. And if we look at around the 1990 to 1995 time point, we see what's known as transcription-mediated amplification. So this is when the development and patent took place. And this is a process in which most detection platforms are beginning to shift their attention towards. So again, as a laboratory, we also have to be always have to be mindful that not all detection assays are the same. As we walk through the detection timeline, we saw that there are differences in time to result that exist. Some tests can take as long as 45 to 55 hours to run. But if we look at newer methods, we can provide results in as little as 12 hours. Some of the impacts that a laboratory could face with these differences in time to result is the difference that 2 to 4 days in turnaround time could do for an operation. If we think about inventory, efficiency, and just getting products in and out and not having to store it over time. There's also differences in accuracy and detection levels that exist. Some technologies are not sensitive enough to detect low-level contamination. Some assays are also not specific enough to detect only the targeted pathogen, and that speaks back to the validation studies that must be implemented by test kit or platform manufacturers. There must be evidence that specificity or inclusivity and specificity or exclusivity are present. You want to be able to mitigate the likelihood of identifying false negatives. So when you identify a positive, are you truly acted on a true positive? Is your test allowing potential positives to remain undetected? And lastly, there are differences in manual steps which are required. As the number of manual steps increase, the potential for error also increases. If you have to do several steps for upfront purification, you're taking samples, spinning down, removing supernatant, adding different chemicals to inhibit growth or to prevent chelating agents, there's a likelihood that an error may take place somewhere down the line. So in identifying an accurate detection assay for yourself, the confidence has to also be present. So how confident are you that the test is being performed consistently and accurately? Again, the robustness of this platform. Will it be able to successfully produce the output that's required for my laboratory sampling needs? So one other thing to take into consideration are the different molecular targets that exist in different matrices and the impact that these targets can place on assay results. As we look at the top half of this schematic, we see that the blue circles represent the target organism. There will be some sort of step present in which lysis may occur. And this target organism is going to be allowed the opportunity to grow through an enrichment step. We want to make sure that the limit of detection is achieved or at least exceeded so that enough nucleic acid may be present for a reaction to successfully and accurately identify. So after a bacterial cell or the target organism cell is lysed, nucleic acid is released, and certain platforms can either look for an RNA target or a DNA target. From this picture, it depicts that an RNA target may be the best identifier due to the large amounts of copies that are present. When a bacterial cell lyses, the amount of RNA, depending on the type, can present either 5,000 to 50,000 copies. When that same cell lyses, the amount of DNA target that's present is approximately 5 copies. So if the right platform of choice for your laboratory was that which targeted RNA, you have a likelihood of achieving faster enrichment times, and you also would have the likelihood of achieving that limit of detection. So a case example that I would like to walk through as one of our takeaways will be that of a false negative or method deviation. If we think back to the validation and verification examples, these validations are performed on specific sample sizes. So if a platform of interest reports that they are validated for a sample size of 25 grams, and you're interested in testing 375 grams, the claims to that validation cannot be carried over because there's now a variation in the sampling parameters. And then I'll lastly touch on a real-world example of how this could serve detrimental in the food industry. So let's take into consideration the potentiality of producing a false negative result. This could be the result of a method deviation, and if a false negative is released into manufacturing or placed onto shelves in the grocery store, that leads to the possibility of implementing a product recall. So these false negatives can come as a change in the sample preparation, handling, or other method parameters that renders the test method unfit for purpose. So again, let's take the example that we're doing a composite run, which consists of a 25-gram sample. The observations are that the technician sampling performs this testing at 375 grams after 24 hours of enrichment. The reality of this is that the method is validated at 24 hours, but is validated at a 25-gram sample size. And this is where the method deviation comes into play. Remember that a validation is only specific within a particular food category and for the sampling parameters that have been implemented. So because the technician stepped outside of the sampling parameters, they have now deviated from the method, and there's a high risk of producing a false negative. The false negative is a result of the CFU falling below the LOD at 24 hours. And next, I'll show you a visual representation of that. So here we have an example of a 25-gram sample on the top left, and on the bottom left, we have what will represent a 375-gram sample. We'll take 1 milliliter of the enriched sample after it's been incubated for 12 hours, and the test tube adjacent to the arrow indicates the amount of organism that could potentially be present after this enrichment time. Each sample would then be transferred to specified volume, transferred to the detection method of choice, and the reaction would take place. After the reaction is finished, the results will be produced, and what will be identified is that at the 25-gram sample, we've fallen above the limit of detection and produced a positive result. The 375-gram sample encountered the same enrichment parameters, the same sample handling procedures, yet the limit of detection fell below the level necessary, and as a result produced a false negative for what truly should have been a positive result. And this is why it's critical that when a laboratory selects a platform and that platform relays claims of validation, that it's understood what sampling parameters were implemented for the validation. So in an attempt to successfully identify these organisms or pathogens in such difficult matrices, there are several things that must be taken into consideration. But I think the greatest takeaways from this presentation have been that the method of choice must be robust. And it has to be validated, and most importantly, it should be fit for purpose use. In essence, can the target organism be detected in the presence of the matrix for this particular method? So in the Customer Applications Laboratory here at ROCA, we have the Atlas. Presented before you are a list of matrices that have successfully been analyzed, utilizing integrated target capture. Several difficult matrices are mentioned here. We have egg products, and egg contains albumin, which has served inhibitory to certain PCR procedures. Spices have successfully been identified, the antimicrobial properties that they possess. We have fruits and vegetables. The presence of polysaccharides have not served challenging. And so, again, it's important that you identify the method that is best for your laboratory, and that has also been validated for a wide array of matrices. So thank you for your time and for your attention. If you have any questions or would like any further information, my contact information is on this slide. You can also visit our website, follow us on social media. If you're ever interested in learning more about our technology, you can also come to New Jersey and visit our laboratory here. Thank you. Thank you. Thank you.